Hi class, this is course of genetics, Bio 225, and this is Dr. Lucy Abgaryan. We are going to go over Chapter 5, Part 1 today. Chapter 5 covers genes, linkage, recombination, and mapping on chromosomes. So let's talk about gene linkage and recombination. Genes that are associated together usually are assorted together. However, sometimes linked genes may become separated by recombination. Farther they are from each other, there is better chance of recombination. There are two themes in this chapter. Farther apart two genes are, the greater the probability of recombination, and recombination data can be used to generate maps of relative locations of genes on chromosomes. So let's clarify some terminology. What does it mean when we say assorted together? It means two genes that come together from parents, they go together in offspring. Recombination, what does it mean? It comes from recombining of DNA, which means that sister chromosomes are being swapped, and we talked about it during the last chapter. If you need a reminder, please refer to the last class material. So let's learn new terminology. Synthetic genes. Those are the genes located on the same chromosomes. Let's look at an example of two X-linked genes in Drosophila with recessive alleles. So we have W plus, which is red eyes, and W, white eyes. We have Y plus, which is brown body, and Y, that is yellow body. So let's look at the situation here. So female parent has white eye and brown body on one X, and also white eye and brown body in second X chromosome. And male parent has red eye and yellow body on X chromosome and also Y chromosome. So in this case, the female offspring, will get either X chromosome from mother, in which case it's the same, so it's WY plus, and whatever chromosome, one chromosome, X chromosome, that has the father, which is W plus Y. However, the male offspring will get one of the chromosomes from mother, X chromosomes from mother, which is in this case the same WY plus, and it will get the Y chromosome from father, which has nothing to do with these properties. Okay, now let's look at F2, second progeny. As we remember, these properties were coming from mother, and let's look at males at F2, second progeny. And as we remember from previous slide, mother had WY plus W plus Y, which we see here that the progeny gets either WY plus or W plus Y. And when genes segregate together, the probability of getting any of these four combinations is 1 to 1 to 1 to 1. However, when we look here, we see that the probability is 99% and 1%, and that is due to recombination. Now let's talk about this in more detailed way. So from original Drosophila slide, we have that F1 female progeny has WY plus W plus Y. Let's think if they segregate separately. What are the probabilities of having different combinations? So we have W, Y plus, we have W plus, and Y. So we can have W, we can have W, W plus, W, Y. We can have Y plus, W, and W plus, and we can have Y plus Y. And probability of each of these ones is 25%. 25%. But since they are segregating together, the probability is 1 to 1. So it's 100%. However, as we saw in a previous slide, there is a 99% probability, and there is 1% probability of W plus Y plus and also WY, which means that there is 1% chance of recombination, where there is part of the chromosome that is being swapped and recombined from somewhere else. So here is more civilized explanation of what we have talked and more detailed. So cross series A, so we have parental WY plus, WY plus for female parent, and W plus Y and Y for male parent. So since this is X chromosome related, then the offspring at F1 the female will get one of the X chromosomes from mother, which will be WY plus in either case, and one will get one set from father, which was W plus Y, and then male offspring will get WY plus from mother only. And then F2 male will get WY plus from mother and W plus Y again from mother. And this there is a 99% of chance for parental um, appearance in F2 male offspring. There is also a chance of about 1% chance of recombinant situation where we will see WY or W plus Y plus. This is the situation when the chromosome is being swapped from sister chromosome. A fragment is being swapped from sister chromosome, and we are seeing WY or W plus Y. And then the cross series B is showing the same situation, the same outcome with different situation, where we see W plus, Y plus for female, either X chromosome carries the same, and then WY for male, and then on the end the same logic goes down the stream, and then we are getting 99% of parental and 1% of recombinant. So here is an example, experimental example. So where we have BC plus female parent and B plus C female male parent. And if there is no recombination, we would expect to see offspring of BC plus B plus C However, we see we don't see that, and there is a which means there is a recombination, and below is the calculation of parental classes and recombination classes, which come to approximately 77% of parental class and 25% of recombination class. So deviations from 1 to 1 to 1 to 1 ratios can represent chance events or linkage. There is statistical model called chi-square that also known as goodness of fit between observed and expected values. So basically we can use this model to hypothesize whether it is relevant to linkage or not. So null hypothesis would be observed values that are not different from expected values. In linkage studies, the null hypothesis is no linkage. If genes are really linked, expect 1-1-1-1 ratio in F2 progeny. Chi-square test can reject the null hypothesis, but it cannot prove a hypothesis. So we cannot say anything behind just rejection of the hypothesis. In order to be able to apply chi-square test, we need some data. The data that we will need to use should come from breeding experiments. We will need the total number of progeny, how many classes of progeny were there, number of offspring observed in each class. So we will do the calculation and the number of offspring expected in each class if there is no linkage, 1-to-1 to 1-to-1 segregation. So here is the formula for chi-square calculation. It's the sum of the number of observed minus number of expected in square divided by number of expected, which also is the O minus E in square divided by E. Consider degrees of freedom, df, in the experiment. Degrees of freedom are the number of classes minus 1. Also that we need to determine the p-value using chi-square value and degrees of freedom. The probability that the deviation from expected numbers had occurred by chance. You can use for this calculation table 5.1, which is in your book. So here is an example with real data in it. So we have progeny, big A, big B, little a, little b, big A, little b, little a, big B, and then the experiment 1, all the data points, experiment 2, all the data points. All we need to do just to put the numbers in our chi-square formula. For experiment 1 we get 2.88, and for experiment 2 we get 5.76. So the p-value should be the value to consider when we are rejecting or accepting the null hypothesis. In classical case, p-value for the cutoff should be 0.05, and here everything in yellow region is where we are rejecting null hypothesis, and everything in the white region is where we are accepting the null hypothesis. So in 1909, Franz Janssen observed chiasmata at chromosomes during prophase of meiosis 1. Chiasmata is the region of chromosomes where two chromatids get together, two of sister chromosomes. And then Morgan suggested that the chiasmatas where the sites or chromosome breakage and exchange takes place. So Creighton and McClintock and Sturm in 1931 found the direct evidence that genetic recombination depends on reciprocal exchange of chromosomes. and there are ways to identify specific chromosomes by physical markers such as dyes or genetic markers. So here is the evidence that recombination results from reciprocal exchanges between homologous chromosomes. So there are these two parental chromosomes above. One has a discontinuity, the top one, and the bottom one has a portion from Y chromosome. So if we look at the left-hand side and no crossing over takes place, then we will just have the defected or the discontinuity and Y chromosome portion um, containing chromosome just going down the line to offspring. However, on the right hand side, we can see when the crossing over takes place, then we are having the situation with the recombinant chromosome. So here is the graphical representation of recombination during meiosis 1, visualized by light microscopy. And so there is the early prophase, where the duplicated chromosome homologs can be observed, and then during the liptotin and zygotin, which we have talked already about these stages of meiosis, these chromosomes come very close to each other, and then in the diplotin stage we are seeing the charismata, how the charismata takes place, and the state of the crossing over of chromosomes. And then here is a continuation of meiosis 1, visualized under light microscopy. Here is the terminalization movement of charismata. So when the exchange, the crossing over has happened, and then two chromosomes are being separated, then there is the anaphase, where chromosome separation occurs after chiasmata reach the telomerase, and then two recombinant and two parental gametes are produced. And here are the haploid products, 4 haploid products. So it was reasonable to assume that further away the genes are from each other, the chances of recombination are higher. So Startwind proposed that recombination frequencies could be used as a measure of physical distance between two linked genes. So the proposal was that 1% recombination equals 1 RF equals 1 MAP unit, which was called 1 centimorgan, and this is the mapping unit for gene linkage. So let's summarize what we have talked about. So for linked genes must be synthetic and sufficiently close together, or the same chromosome on the same chromosome, so that they do not assort independently. This is for the linked genes, and for unlinked genes, they occur when either when genes are on different chromosomes or when they are sufficiently far apart on the same chromosome. At next lecture, we will continue about talking about this chapter, and particularly we will talk about gene mapping.